and that they carry on her genes. While for men, this is less important because they can just, churn out more and increase their fitness that way. For males in the animal kingdom. And that is this big discrepancy that has led Charles Darwin to formulate, of course, sexual selection. You're all familiar with this, where he says, well, female choice and male mate competition is one of the guiding principles of evolution and the evolution of and the maintenance of variance of traits. So, and in theory, sexual selection is just this. We have the within sex, so from a male perspective, we have competition among males for access to females, because that is the limiting factor for them. So the more females they gain access to, and the more female they can make offspring with, they can also increase their fitness. However, for females, it is more important to choose, and female mate choice is one of the most interesting principles, I think, in sexual selection. And that brings me to extra pair offspring, which is my main topic. So extra pair offspring is when a bird clutch, like a brood that is being cared for by a socially monogamous bird species, by a pair, a male and a female, who both feed or incubate the young. When sometimes one of the young is not fathered by the male that attends the nest. And that is an extra pair offspring. That means the female has gone astray and has fathered offspring without him. 3 with him and 1 without him. And that has very sex-specific consequences, because for a male, he can have his own clutch that he cares for and provides parental care to, but he can have an extra one and another one. While for the female, this is not the case. She can either have 4 offspring with her social male, or she can have 3 offspring with her social male and 1 other. So it's an or choice for her. Well, for him, it's an and choice. So in males, it's additive. In females, it's a replacement. And all she replaces when she engages in extra pair behavior is the paternal genome, of course. So it's a simple replacement. Everything else is being kept the same. And that is what I want to talk about today. Why do females do this? So she just replaces this. And that's, of course, the female choice principle that Darwin told us. And the nice thing about extra pair reproduction, and the reason why I like it so much, it's excellent for testing sexual selection theory, because it really shows choice. So the female has to make a choice between the within pair father and the extra pair father. And she's made a choice in this picture of replacing one of them. So it's actually a beautiful natural experiment to test several predictions of sexual selection, because the environmental variables are kept stable. All chicks receive the same care, have the same environment, and all of these things are kept the same. It's just the genes that are different. It's beautiful. Beautiful. Yeah, it's very nice, actually. So, and I study this in the house sparrows. House sparrows are socially monogamous, and they have sexually, the sexes differ. So the male has a black bib here, and is in general a bit more ornamented than the female. All of you will know house sparrows, I hope. They live with humans in farms and barns and follow humans around. They're socially monogamous. About 25% of the broods do contain extra pair offspring. They have biparental care, and long-term pair bonds. So males and females stay together, and both of them really care for the young, and there's not that much difference between male and female care. So the males actually are quite invested in their social broods. And another nice thing is they have between 2 and 4 broods per year. And that's quite nice because it gives you repeated measures, even if you only measure something in 1 year. And on average, they have 4 eggs. And I study house sparrows in Lundy Island. And I just want to show you here, before I introduce Lundy, actually, the percentage of broods with extra pair on Lundy varies between 0.2 and partly it even goes, it's like that every second brood has an extra pair offspring, which is quite neat. So this is a really nice bird species to study this in, I think. So, and I just very briefly, I think you all know this, but I'll just briefly go through this because I think it highlights some of the problems when we study extra pair paternity about how we do this. Of course, we do paternity by exclusion. So in our study up to now, we've always used microsatellites. So it's a bit old-fashioned, but it's quick and cheap, and it works very well. So what we do is, of course, we genotype the mother, we genotype the offspring, then we look, what overlaps between mother and offspring. So the mother is homozygous on this locus here. So the 100 base pair locus, obviously the offspring got from allele, obviously the offspring got from the mother on this locus. But then there's another one, and then we compare that with his social dead. And if his social dead doesn't have that allele, then obviously this offspring is an extra pair offspring and has been fathered by somebody else. Because that allele, this 104 base pair long allele, needs to come from somewhere, right? However, this is where many studies stop. And the problem is really finding out who is the extra pair father. It's usually fairly easy to kind of, genotype all the males you can catch, and then you'll find one that maybe has also this allele, but you can never be 100% sure that this is the father. There might be a bird that fathered this offspring and then flew away again. You never sampled it. So there's a big sampling problem. So how can we ensure that we sample all potential males? And then if you have a large number of markers, of course, you can exclude many males from being the father, and then usually only one remains a possible genetic father. But you can only do this if you sample all potential males. And the way I do this is I sample on Lundy Island. And that is, as you can see here, this rocky outcrop, and there's not much habitat for sparrows on this island. And if you look in the middle, you will see a little village. And in that village is one farm, exactly, with one barn and two stables. And that is where the sparrows live. And they don't go anywhere else on the island. And that setup allows me to really catch all sparrows every year, because it's so small that with enough effort, we actually get everybody. We also know, we've put up nest boxes, obviously, and we also know where they breed if they don't breed in the nest box. And we also look for other wild locations, although there's not so many. They get very used to nest boxes. It's quite neat. Lundy is located in the Bristol Channel. So it's west of Wales and Devon here. And it's about 19 kilometers away from the coast. And that is far away for sparrows not being able to immigrate. So I've looked at my genotypes, and I can tell you we've had probably less than 10 immigrants in the last 20 years. We had a few more because that was an experiment, but there's very, very few that come into the island and settle. And, so we can be fairly sure that there's no unknown male that kind of just, flew away, flew there, fathered an offspring and then disappeared. What we cannot be so 100% sure of is that none leaves, because we do catch and we monitor the closest mainland, and every now and then some birds pop up there, but I think they're mainly blown away, and it's not very many. But I guess my point is there's some gene flow. So yeah, we have nest boxes, and every bird gets 3 color rings and a metal ring so that they are individually recognizable. They also have an RFID tag. Birds feed their young in the nest boxes, so we can take videos and measure their parental care very well. We can also observe all their broods by just, monitoring the nest boxes in a standardized way all through summer. In the off season, we also go and we catch birds in the winter with mist nets, just to make sure we have everybody, and we have, most importantly, not only all the measurements of them, but also blood samples. So we take blood samples of every chick that is born on Lundy. Most of them we get in the nest. Very, very rarely we lose some because they bred after our students have left or before, but we usually catch them in the winter or because they're an immigrant. But we have generally more than 99% of those that have been bred are also genotyped, and of these 96% have been assigned parentage. It's not perfect. The reason for that is that we make field work mistakes and we make lab work mistakes. And that is actually, it's one of the drawbacks when you work on such a highly systematic study, because if you take multiple blood samples from the same bird across sometimes 10 years, you will be able to trace errors back. So I can actually really usually assign which errors come from where, when people have swapped blood samples or mislabeled them, or when there was like a pipetting error in the lab. So with enough data, you can actually infer that. It's quite interesting, but it's also a bit scary, because if you don't doubly genotype, you will never know. Anyways, what I've done with this data is I've put together a pedigree, which is fairly complete, at least from the year 2000 onwards, with some data from starting even in 1987, because BTO ringers have given us data, and some people even have taken blood samples back then, and we were able to regenotype them. But the reliable data starts in 2000. It's more than 20 generations. As I said, we have less than 50 immigrants. 41 of them are an experiment, and 10 or less are natural immigrants. And we know exact paternities. But another good thing about this system is we also know precise long-term fitness. Not only can I calculate lifetime offspring, I can also calculate the number of lifetime recruits, where recruits are defined as birds that actually not only survive, but also reproduce themselves. And what we can also do is we can calculate lineages and lineage survival. And one of my students has done that. She's looked at, as a way of verifying that the number of recruits is a very good fitness estimate. She calculated the correlation between the number of recruits, the number of lifetime recruits produced, with the expected genetic contributions, and that correlation was very high. And these are, here in the plot, you see on the y-axis the expected genetic frequencies. These are the contributions of lineages over the years and how they vary. And you can see there's quite some variance there. But I guess the point of this slide is to convince you that we have very precise fitness estimates, and very unusual for a wild system, I think. But let's get back to extra pair reproduction and focus on the fact that this is a natural experiment and that we can easily compare female choice for males. So the question is, why do females cheat? And as I've elaborated before, is unlike males, they do not gain fitness other than if they make a wise choice. They only swap the paternal DNA. So if we go back to Darwin and sexual selection, we can come up with predictions for this. And the very easy predictions from sexual selections are: males gain direct fitness benefits if they engage in extra pair behavior. The second is females pay direct costs by means of sexually transmitted diseases, or because males might retaliate. Females are also predicted by choice to gain indirect benefits, such as the genes that the extra pair father leaves them with in their offspring, and as such, a female will also gain fitness. And similarly, she can gain fitness by choosing cleverly, not only best genes, but choosing those genes that are best fit for hers. And then another idea is that females may also gain a fertility insurance, although that is a bit of a difficult to test theory, and those studies that did test it found that it's likely not the case. Anyways, but if we take these predictions, we can translate them to very solid and robust predictions for Lundy, which is that extra pair fathers have a higher fitness. Fathers do not care well for broods that have extra pair offspring. The extra pair offspring are of higher quality than the half siblings, and the extra pair fathers are of higher quality than the social fathers. And we can test all these in the Lundy data. So I'll walk you through all these briefly, some not so briefly. So the first prediction, extra pair males have higher fitness than monogamous males. So what we did is we looked through our data set, we picked out all males that produced any extra pair offspring and thought these are not monogamous. And then I picked out males that never produced any extra pair offspring. And that is something that you cannot do in any other data set than an insular one, where you're really sure that there's nothing else. Because I know that these males never produced any extra pair offspring because none of the birds that we sampled had these as a father. I cannot know that they've never copulated extra pair, however. Anyways, of those males that produced any extra pair, we also separated out what we call floaters. These are males that only produced extra pair offspring, but never held a social brood themselves. I think it's important to classify them out because they usually are younger males that then die very early, so they will have a lower fitness. As response variables, I use the lifetime number of offspring, which is just offspring produced, and then the number of recruits. These are those offspring that then go on and have own offspring. And what you can see here is if the Zoom thing is not in the way, which it is for me. What you can see here is the sample size. We have 192 extra pair fathers, 139 monogamous fathers. It's over a lifetime, right? And 65 floaters. And you can see that indeed extra pair fathers have a higher fitness when we look at lifetime offspring number, but also when we look at the lifetime number of recruits, that pattern stays the same and the floaters have the lowest number. So, and it's quite interesting. So the monogamous ones have less than one and the extra pair ones have a bit higher than one lifetime recruit. So we can do a tick here and then we can look at the next one. The next one I want to go into a bit more detail. The predictions is that fathers do not care very well for broods that hold extra pair offspring. And let's have a bit of a closer look at this. So the idea is that if a father cares for a brood of 4 and he sired all of them, then he pays the costs in parental care for 4 chicks, but he also gains the benefits, fitness benefits of 4 chicks. However, if one of these chicks is not his, he still feeds it, right? Because, well, it's in the brood, right? So he has to work more than the benefits he'll get out of. And because of that, the prediction is, if a father would know that, a social male, it would reduce its parental care to those broods that have extra pair offspring. And this is what we've done. We've taken videos of all these nest boxes and birds flying up and down. I've had a lot of students sit and watch these videos and decode them. And then with this data, we can look at whether cuckolded fathers will reduce paternal care. And the prediction is, of course, that the less paternal care, the more extra pair paternity, the other way around. This, about 25 years ago, this was very fancy and people really liked to study this because it was so easy to do. However, there's a couple of problems with this, and it does not show causality, because poor males may prompt females to cheat, or poor males may just be of poor quality and therefore may not be able to care so well for their brood. So they're not necessarily, there might be a relationship, but it not necessarily needs to be causal. People done it anyways, and this has led to Bart Kempenaars from Max Planck and Ben Sheldon from Oxford to write this series of commentaries, which were really scathing, I think, to comment on this. And they wrote, and I'll just quote, "As a consequence, we argue that it's currently impossible to predict what relationship may be uncovered between paternity and paternal care in studies relying on correlations. Little is to be achieved by reporting such relationships." So they were really, really harsh with this. And that was in 1997, right? But they're saying it's just inherently impossible. Did not stop people from doing this, obviously, which just made them even more angry. And they wrote another, another comment, which is called Confounded Correlations. And he said, "We do not agree. In our view, these studies provide no useful information about causality, and we hold this view whether the next 50 studies in this area produce positive, negative, or any mixture of relationships." It's really harsh, I think. But as you all know, science is not easily demotivated, so people went on and studied this in large numbers. And until in 2013, Ashley Griffin from Oxford also managed to put together a nice meta-analysis, and the result you're seeing here. So on what she's shown here in many bird, but also fish and mammals, that indeed overall, if you do a meta-analysis, you find that yes, there's an effect, and that cuckolded fathers reduce paternal care. Of course, this is between species and not within species. So, well, and this is a really nice meta-analysis, but the causality problem remains. That doesn't go away, right? So poor males may prompt females to cheat, and it's not showing any adaptive response. So what I did is then I looked back at these 1997 papers, and what they wrote is they had actually a suggestion of how to solve this. And they wrote an alternative approach, which has proven fruitful, is to restrict comparisons of paternity-paternal care to successive breeding attempts by the same pair. So what they're saying is you can do that in repeated observations on the same male. And the idea here is that if one male that is rubbish at paternal care and whose female produces a lot of extra pair offspring, if this one would be paired with a female that produces less extra pair offspring, or if the female would just produce less extra pair offspring in a second brood, would he then, change how much paternal care he gives, or would he stay the same, and the same with a higher quality male? And this would be a much better test. It still wouldn't be an experiment, but it would be much better. And this is what they're saying, and that makes sense. So what I did then is I went, oh yeah, you can do this actually in mixed models statistically, which is quite nice, using the within subject centering methods, but this is very data hungry. So what I did is I went back to the meta-analysis, had a look how many of these analyses actually used repeated measures, and there were not many. And then there's one that wasn't in the analysis actually, that came out a bit later. And this is the sample size. So the highest is 15, but it's more, well, around 10. And that is not large enough to do a proper analysis. So on the Lundy data, we actually have much better data. So what we do have is over 8 years, when I wrote that paper, we had 863 repeated observations on 186 males for incubation visits. And for provisioning visits, we had 1,430 repeated observations on 207 males. So that is significantly more than 17. And I thought our data set might prove a really good, good opportunity to test this. And I just throw this in because I know Rita is interested. So we also now use machine learning. It used to be that students would just watch the videos and just go through every, frame and look at how often the birds will provide care. But now we also use machine learning, and that works quite well, and it saves us a lot of time, which is a recent change. So we have much more data than that now actually available if anybody's interested in that. Anyways, so what I use the within subject centering technique. Martijn van der Poel wrote this up very nicely in Animal Behavior. What it allows you is to distinguish the main slope across the population from the within individual slope if you have repeated measures there, which is quite nice. It's a bit like random slopes. You can also estimate that on a random effect term if you want. It's a different method, but this works quite well. And when I've done this, I didn't find any effects in the incubation behavior, but when I looked at the provisioning behavior, I did find a strong negative effect. So the higher the extra pair frequency in the nest, the lower the provisioning visit rate for the male. So males seem to adjust paternal care to extra pair rate, and that was a within individual effect, which was quite cool. However, this is still not all, because this effect, because males, it can be repeated measures when a male is with the same female and the female changes, and this is what you really want to know, or it can be when they switch partners. And to find that out, so you actually want to do an iteration of this within subject centering thing here to find out whether this slope is what direction it is. And I've done that, and you can do it. I can talk to you about the math if you want to, but it's quite neat actually. And when you do this, what I found out is here, this is the within male provisioning. It's not statistically significant anymore because it overlaps 0. The between male one is not statistically significant anymore because it overlaps 0. But what is significant and negative is the between pair one, within male and between pair. And then I have my covariates here. What does that actually mean? Because this is a lot, right? So it means when a male stays with the same female and she adjusts her extra pair, the number of extra pair in the nest, he does not change. He does not change. So it's a hedge, he decides on how much parental care he will give to that female, and he'll give that reliably in every brood he has with her. However, if he switches pairs, if she dies or if they have a divorce or whatever, then he adjusts how much pair he gives, and that depends on how much extra pair offspring this female produces. So he actually does not adjust to the number of extra pair offspring in the nest, but rather to the identity of the female. And that is actually supported by, we've done another analysis where we looked at whether males provide different care to broods that are not related to them. We also do cross-fostering quite a lot in this population. So what we could do is we could look at the visits per hour, male parental care visits per hour, to broods that he was fully related to, that he was partially related to, or that he was unrelated to. And as you can see here, absolutely no difference. So then no, they cannot recognize kin in their nest. So this all makes sense. So yes, females pay costs. There is a cost if they have more extra pair they will find males that produce less offspring to them than they would be able to. But it's not because fathers adaptively respond. It's they adaptively respond to the social identity of the female. And then the next ones are a bit easier to look at is female gain indirect benefits. And the prediction from that is offspring, extra pair offspring are of higher quality than the half siblings. Let's have a look at that. Idea is this offspring, the green one, is better than these. And we can look at that, of course, with the estimated probability to hatch, and then later on also with, number of records and yada yada. However, we're not only comparing the extra pair offspring with the within pair offspring, we're also comparing them to offspring that come from broods that have no extra pair. Because the assumption is always that females go and get better quality genes for the extra pair. What if they already have a really high quality social male? Then they maybe don't have to do that. And this is why we separated them out. However, you don't find any difference between these. So, and that was the same for nestling survival, for recruitment to the breeding population, for lifetime reproductive outputs. So none of these extra pair offspring were of better, if any, there were worse condition than the within pair offspring. So this doesn't work, clearly. And then we went on to test whether fathers are of higher quality than the social fathers. And we can do a really nice pair test, because this is ultimately what Darwin predicted when he said females make a choice, right? In this case, a female says, no, I don't want, for my fourth offspring, I don't want the genes of my social partner. I want the genes of the extra pair partner. And no matter the costs, I'll do it. So, well, there must be something that she gains from him. So, but we did not find any differences on Lundy other than that the extra pair fathers were older, but they were not better. But what we then did is we went and done a meta-analysis where we looked for other studies that have looked at that. And we found between 32 and 40 studies. And when we looked at the outcome, we find that neither genetic similarity to the focal female, nor any body size indicators, nor any sexual ornament size were statistically significant. So the only difference we found where extra pair males were better was age. So extra pair, older males were better able to secure extra pair paternities. So this was a bit of a, well, that was a bit frustrating. So we still don't know why females do this, because they only pay costs, but they don't gain any benefits. So, well, so we need to look at that sexual selection theory in a bit more detail. And lucky for us, Ronald Fisher came along with the modern synthesis to rescue us. So people started thinking a bit more about quantitative genetics, although Halliday suggested the sexually antagonistic pleiotropy hypothesis in 1987. Nobody really considered it as much until 10 years ago, maybe, where it started to kind of pop up. So we have two hypotheses. One is the intrasexual antagonistic pleiotropy hypothesis, and the other one is the sexually antagonistic pleiotropy hypothesis. Both of them are a mouthful, and I'll talk you through them. So in the first one, the idea is that a trait is selected for in one context, but not in the other. So say in females have a high sex drive, and that is selected for in a within pair context because that gives her a higher fitness, right? But then in an extra pair context, it may not be beneficial, but if the within pair context gives her so much more fitness, then it can just, hang on. But you need this sort of genetic correlation between both traits. But if it's the same trait, and if it's, coded for by the same gene, because it's just, I don't know, sex drive or just a higher propensity to engage in copulations per se, then this is possible.
See.
The other theory, the sexually antagonistic pleiotropy, suggests that the same genes in males and females code for that behavior, but it's adaptive in males, but not in females. So if it's super adaptive for males and males really gain tons of fitness benefits from this, then the female trait, even if it is maladaptive, might actually evolve together. But for this, you would expect antagonistic genetic correlation, negative, also a genetic correlation between the sexes and antagonistic selection pressures. So I want to focus now first on the intrasexual antagonistic pleiotropy and look at what kind of predictions we can come up with this. And the easiest is, of course, females that copulate more with within pair partners will also copulate more with extra pair partners, or females that copulate more in general will also have more extra pair offspring. So this has been tested. Wolfgang Forsmeyer, my colleague at Max Planck, has tested that in zebra finches. And well, he's found some results that may be supported or maybe not, but there's some caveats to the whole thing, in that the social identity of the female and the male might be more important. And the problem testing this in captivity in a zebrafinch setup is that you're limited by how many males you can provide a female to choose from. It's usually choice experiments where a female sits in one chamber and is presented by two males. Another problem by other studies that try to look at that is that they're measuring the outcome, the number of extra pair, but not the actual behavior. So what I thought we could do to kind of make this better is also go to captivity. So I have sparrows in captivity and actually look at them in a group. So we keep them, sparrows are kept in groups, not in pairs like zebra finches. And you can actually, when you give them rings, you can follow them around and you can actually quantify how often they copulate, individuals copulate, and with whom. It's tedious, but I've had a really good student who did that. So I'm testing the prediction that females that copulate more within pair also copulate more extra pair. And here you see some of the results. This is the number of total copulations that females do, and it's plotted against the number of extra pair offspring that result from this. And we did not find anything. And it doesn't really matter what you plot. There's just no relationship. And I found that very frustrating. However, Antje also found, the student who did this, that females actually only copulate when they solicitate the copulation. So here in this plot, you see on the y-axis the probability of copulation. And on the x-axis is whether a female solicited, on the right, a copulation or not solicited a copulation. You also see the sample sizes here. And the black dots are extra pair copulations. And you can see on the left, the unsolicited black dots, females are very, very, very rarely only copulate extra pair when they did not make that choice. So there is a strong element of choice, but we could not really find this link, this particular link here. And what made me think that maybe, again, the social element is more important is, first of all, this finding that I found that males really look at the social identity, but also the zebra finch study suggested that there's a strong social element going in. And I think this is what has come up in the last 5 years that we really kind of ignore when we look at our classical theories, and we ignore the concept of individuality and how they are connected. And that really brings me to, well, social network theory. I rather call it social modulation theory. And I also don't really want to put Mark Zuckerberg on par with Darwin or Fisher. So I'd rather use that depiction. Anyways, so the idea that social networks are important and that opportunity hypothesis, and the idea is that if there are more, if a female has more associates, she has more opportunity to actually assess her choice and make a better choice and have a higher fitness. And if she seeks out more social associates, she also has more opportunity to copulate extra pair. So overall, this idea is that this social network of associates informs mate choice and modulates the pleiotropy hypothesis, really. So, and this is what we set out to do. So what we do on Lundi is, you can see this here. So this black square on the ground is an antenna that registers RFID tags that these birds have all implanted. And the beauty of the Lundi system is that we have a plug where we can just plug it in and it just runs forever, you know. So as long as I have somebody who refills the feeder here, there's food in here, I can actually collect data all year long without actually doing much, which is quite neat. And that's what we've done. And then you have this presence data, and from that presence data, you kind of put together social networks, right? Where you have, well, these two are probably associated, and these two clearly are associated, and these are all associated. And when you do this, you come up with a social network. And when you have that, you can actually calculate all sorts of sociality traits. And this is a bit of jargon, unfortunately, that I have to jump on you. And in social network lingo, the degree trait means the number of connected individuals. Strength is also the number of connected individuals, but weighted by the intensity, by how often they associate with each other. And then you have these several concepts that are called centrality, closeness, betweenness, which kind of talk about the connection to the population. So birds that are more central in the network and have more connections probably score higher than those that are at the end and very singular. There's a problem that comes up when you do this, because we have, of course, the data is blind to what they actually do. We also did some video analysis and built networks with that, but we really wanted to get a good picture of what these sort of RFID readers get. And the problem you have is, how do you define a social association? If one is a bird that over time stays at the feeder like this, 2 is a bird that went to the feeder, flew away quickly, then came back, 3 is a bird that stayed here. So are all birds together associated, even though this bird never, the one bird never, actually spent time at the feeder together with bird 4? This is the gambit of the group. Many studies assume that because they think there's flocking going on. However, in our bird population, what we found is if we use the gambit of a group, is we get really unrealistically large networks, and like the whole flock is the whole population is a group or a network. Another approach is using a time overlap between birds. So if this bird overlaps with that, it's associated. This bird's overlap with that. So 1 overlaps with 2, but not with 3. So only 1 and 2 are associated, and 2 overlaps with 3 and 1. So 2 is associated with 1 and 3, and so on. But if you do this, and if you have like our birds, like constantly replenishing birds at the feeder, you get very long linear networks, which is also not really realistic. So what our students have done is they made this definition about arriving. So if you have a bird at a feeder and a second bird arrives when this one's already there, and this one doesn't fly away right away, then you have an honest interaction, and we're validating that with video analysis. So these are the type of interactions we're using. So it's not a simple have been there on the same day. So it's quite an elaborate mechanism of calculating associations. But we can do that because we have enough data, I think. So we can be very very picky in how accurately we define interactions. And what we've done then is we've looked at repeatability of these traits first. And I looked at repeatability in captivity, and on Lundi, Mireia actually did that. She was a PhD student who came to us for, I think, half a year, and she did that. And so we done this in A, you can see Lundi. So the red ones are actually the ones you want to be looking at. The other ones are the randomizations to compare against. So you can see that on Lundi, we have reasonably high repeatability. Zevies, this is captive population, just in males is also very high. Silwood is also captive population, also very high. And then what we've done is we've switched the population Silwood up. We mixed up birds. We took old social connections away and put them in different aviaries. And when you measure then up to 3 months after, it takes a while until these kind of networks reoccur actually. So, and then it goes to 0 here after the change in the social environment, but after a while it came back up actually. So it's quite nice. So yeah, the traits build up over time, they can be disrupted, but there was no big differences between males and females, and all traits are repeatable, which is nice. And then Jamie looked at the association between these traits and fitness. And what he found, and he used a measure of derived fitness, which is a bit like which is a contribution to the next generation, the genetic proportional contribution to the next generation. But this also works with a number of lifetime recruits. If you look over lifetime, you find a squared association with centrality and with degree, which indicates there's some stabilizing selection. So the birds that have average sociality have highest fitness, or over lifetime fitness. And oh yeah, this is just to give you an idea of, we had 400,000 visits across 2 winters, about 2,700 visits per bird and 150 birds. But of course, what we really wanted to know is what's with the association between opposite sex members. So what he did is then he calculated how many opposite sex friends, degree, each bird had. And so he only considered opposite sex social associations. He made a new network with only that. And then he looked at annual fitness. And what he found, and he didn't make me a really nice graph. So this is still in the making. So this is very fresh data. He uploaded it to bioRxiv before Christmas, I think. So, but what he finds is that opposite sex degree is actually positively related with annual fitness. So if birds have more opposite sex social associations in the winter, they will have a higher fitness in that year. And that is linear and not squared. So for the opportunity hypothesis, we can say, yes, the more social associates, opposite sex social associates, the higher the fitness. But there's still a number of open questions, I think. The next question is, the more social individuals have extra pair interactions. This is, of course, what we really want to know, right? So let's have a look at the data here. First of all, I find that more social individuals have more mates per year. So they are more switching mates or have more extra pair mates, which is nice. This is Lundy data. However, when you look at the link between opposite sex associates and extra pair offspring, you will find that, sorry, here I'm looking at the opposite sex degree, and there's a slight difference between the sexes. Females have more opposite sex associates than males. We find no effect of age. That is important because when you look at the number of extra pair offspring and the opposite sex degree, you find no relationship, and if any, it's a negative one, which is different from what you would expect. So that was a bit disappointing. Maybe we still don't have enough data, I don't know, but yeah, so this does, I say it predicts, but it does actually not predict well the number of extra pair offspring. So what I've done then is, so we're not so sure about this. What I've done then is I've looked at, I thought maybe we should, do an experiment. And that's what we've done. We went back to the captive birds, and I'm actually quite excited about this. So Mireia has done this, and she's put together, she's looked at females in different aviaries, and she partitioned them into females of high sociality and to females of low sociality. And she did the same for males. And then she put these together in the sense that you had one aviary that had high female, high male, one aviary that had high female, low male, one aviary that had low male and low female sociality, and one aviary that had low female and high male sociality. And it was a bit difficult because you had to, of course, first measure them, and then you have to take out the middle bit and just take the extremes, right? And this is what it looked like then when you put them in and measure them again, but you will still find that the females were quite high in the high groups, as were the males. And the low ones, well, there was one group where the males, it wasn't so good, but the rest was reasonable. And these were put together then, females and males, so that you had like these really extrovert aviaries where they kind of went on and paired and copulated, and then they had these really like introvert ones and mixtures between. And then we measured extra pair copulations and also extra pair offspring. And what I'm showing here is the proportion of the extra pair copulations of the total populations of the individuals in the different treatments. And you have high, 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 low, low, high, and low, low treatment here. And what we actually find is that indeed in the high treatment aviaries, the birds copulated more extra pair than in the low treatment aviaries. And I think that is quite a strong suggestive evidence for this theory. So yeah, we can put a careful tick there, I think, but there's still questions. But I think we can definitely say the social network of associates does drive mate choice and needs to be considered when we look at this. Last bit here is, and I'm not spending a lot of time, this is the sexually antagonistic pleiotropy hypothesis, where you expect a genetic correlation between extra pair reproduction within pair reproduction in males and females, and extra pair reproduction in males and females. Yeah, we put a tick here. The problem with this is evidence for this is very, very scarce. It's actually the only one who did it experimentally was Wolfgang Forstmeier, and he found he did this really interesting thing where he did this really long-term study with his zebra finches for years and years and years, and then he published it because he did find this effect. He published it, and then because Wolfgang is Wolfgang, he's lovely, he's a really good scientist, and then he continued studying it, put all the data together, and then he didn't find the effect. It's like, what is happening? So it kind of changed his conclusions again, and he published both, which I thought was really good. So we don't know, but he also says there are social effects that we should be taking into account and that are not known yet. So there's a lot of open questions, I think, on this. So in the end, why do females cheat? Well, both Fisher and Darwin have given us really good ideas, and we could find support for quite some of these hypotheses, but it's clearly more complicated than that. And we need to take into account social effects. So why do they cheat? Well, the social network of associates modulates mate choice. Antagonistic pleiotropy is very likely important. The importance of the social environment should be considered in studies. And we should also try to measure more the behavior copulations actually instead of the outcome, extra pair offspring, which of course is not easy. And the knowledge of the complete sexual history is important. So if you just do 1 year, it's really likely that you get a bit of a weird effect, and that is maybe not so reliable. This is mostly it. So I have an outlook. So we clearly need more social data. This is what I'm working at. We are working at, my students. We're also working at getting a better measurement of copulations to really quantify that, not only in captivity, but also in wild birds. I also work at senescence and transgenerational aging. And recently we started now to actually sequence the whole lot of Lundi and look at the genomics of individual fitness. And that obviously will kind of also permeate this research topic quite a lot. And I'm looking forward to that. And with this, I want to end. Obviously, this is not my work. There's tons of people involved. And I want to particularly highlight Terry Burke on the top right, who founded the Lundi house sparrow population really, and took me on as a postdoc in 2009, and I've never let go of that. And it seems like I'm taking over the sparrow population now that he's yeah, this is what's happening. It's a bit scary actually, but this is what I'm preparing for and what he's preparing me for. And I also want to mention Jamie Dunning on the top left, who's a current PhD student, very keen on collaborating with people on social networks and extra pair paternity, and he's a very good bird also. If you hang out with him, you always find any good lifers, or at least I will. But there's a ton of other people also who've contributed to this, and I want to thank you for your attention. Thank you very much.